Yes, what we are going through the reproduction of the protistans. It is asexually type and sexually type. Asexual type by budding, spore formation and fission, binary fission or multiple fission. Then we have sexual type, isogamous, anisogamous and oogamous type, where both the gametes are same. Gametes are same. Then it is called isogamous type of sexual reproduction. When both the gametes are not of same shape and size, then this is known as an isogamous type of reproduction. And here the oogamous type, both the gametes are not of same shape and size, but there is a difference from anisogamous and oogamous type. In this case, the female gamete is larger than male gamete and female is sessile or it cannot move. But in this case, they can move in spite of having the difference in the size. So anisogamous and oogamous type. So isogamous type of reproduction, anisotype of reproduction and oogamous type of reproduction, they can be found. So in isogamous type of reproduction, it is found in microcystis. Anisogamous type is found in ceratium and oogamous type is found in plasmodium. So isogamous type, anisogamous type and oogamous type of reproduction takes place in the protista. Now, the whole organisms of kingdom protista can be categorized into different groups, or the kingdom protista includes: number one, dinoflagellates. Number two, diatoms. Number three, euglenoids. Number four, slime molds. And protozoan protist. Dinoflagellates, diatoms, euglenoids, slime molds and protozoan protist. So whole kingdom protista includes dinoflagellates, diatoms, euglenoids, slime molds and protozoan protist. Now we will discuss about first dinoflagellates. So what are dinoflagellates? So kingdom protista is basically involving or containing those types of organisms which are unicellular and eukaryotic. Now first we are going to deal with dinoflagellates. So this group of organism or protistan is golden brown in color. They are golden brown in color. Next characteristic, that they are photosynthetic. That is, they can synthesize their own food. Photosynthetic. photosynthetic. So they can perform photosynthesis and they can manufacture their own food. Now, next thing is they have two flagella. Two flagella. Okay, two flagella. That is why they are known as dinoflagellates. Two flagella. One is longitudinal and one is transverse. That is why they are known as dinoflagellates. So you can have a look on the representative diagram of dinoflagellate. This is transverse flagella and this is longitudinal flagella. And these are the plates. These are the plates. So that is why they are known as longitudinal and transverse, having flagella, dinoflagellates, and they perform photosynthesis. So as they perform photosynthesis, so they must contain some type of photosynthetic organelle, that is chloroplast. So these organisms contain chloroplasts or chromatophores. Chloroplasts or chromatophores and contains photosynthetic pigment. Photosynthetic pigments such as chlorophyll, chlorophyll A, chlorophyll C, alpha carotene and xanthophyll. Xanthophyll. So chlorophyll A, chlorophyll C, alpha carotene and xanthophyll. So with the help of these pigments they perform photosynthesis. Now if they perform photosynthesis, then they must form some product. So the stored food is starch, but in freshwater forms. That is those protistans, those dinoflagellates which are found in fresh waters. Then stored food is oil in case of marine dinoflagellates. If marine dinoflagellates, they have oil as a stored food material. So these dinoflagellates are having two types of flagella, and they are photosynthetic and they are golden brown in color, and they reproduces sexually as well as asexually by spore formation and budding, and sexually with the help of conjugation and with the help of isogamous, anisogamous and oogamous type of reproduction, but they perform usually isogamous type of reproduction. Now we are going towards the next group, that is diatoms. That are diatoms. These diatoms are those protists. These diatoms are those protists comprising of two halves. Comprising of two halves, just like soap case. They comprise of two halves, just like a soap case, and their cellulosic cell wall. They have cellulosic cell wall. That is, the cell wall is impregnated with the, or it is formed with the cellulose. And these are not degenerated because their cell wall contains silica. Their cell wall contains silica, and they have made up of cellulosic cell wall. The cell wall is impregnated with silica. That is why it is not degenerated and forms diatomaceous earth. Diatomaceous earth that can be used for filtration in different types of industries. Then the diatoms are basically made up of two halves, and two halves are known as frustules. These are known as frustules. Then these are performing photosynthesis, and they are going to prepare its own food. So the photosynthetic pigments are chlorophyll A, chlorophyll C, carotene, and fucoxanthin. What is this? Fucoxanthin. So they also perform photosynthesis and manufacture their own food. And diatoms, they have frustules and perform photosynthesis with the help of chlorophyll A, C, and carotene, called fucoxanthin. That is why they are called photosynthetic protista. For example, Pinularia and Melocera. So these are dinoflagellates and diatoms. Now we are heading towards our next type is euglenoids. What are they? Euglenoids. Means euglena like organisms. Euglena like organisms. And more interestingly, they are not having a cell wall. They are devoid of cell wall. Rather than covering with the cell wall, they are covered with a membrane called pellicle. So just before going further, just have a look on the diagram of a euglena. Just an indicative diagram of euglena. This one. It has two types of flagella, one long and one short. This is long flagella This is short flagella. This is pellicle. This is pellicle, and this is known as reservoir. This is reservoir, and this is known as eye spot, and this is cytopharynx. And this is known as cytostome, and there is paramylum granules. These are paramylum granules. These are paramylum granules, that is, this is the stored food material. It is the stored food material in case of euglenoids, because it performs photosynthesis in the absence of light, and it can also perform the type of animal type of nutrition in the absence of light. In the presence of light, it can perform photosynthesis, and in the absence of light, it can perform animal like of nutrition. That is why they are known as connecting link between animal and plants. So these having the paramylum or paramylum granules as stored food. Because they perform photosynthesis with the help of chlorophyll and carotenes, and they do not possess any cell wall or any type of shell. They have pellicle, that is why they can be in the form of slipper, and they can move where they want. So this is the indicative diagram of a euglena and the euglena-like organisms. It moves with the help of flagella, and it performs different types of functions. Now we are going towards, and here this is the cytostome. What is this cytostome? It is comparable to the mouth of the organism, and this is the canal. Cytopharynx is the canal through which food is ingested. So this was the euglenoid or euglena-like organisms, and the famous or the most important member of these euglenoids are euglena. Now heading towards the consumer protist, that is slime molds. That is slime molds. What are these slime molds? These animals are interesting because they contain the characteristics of both fungus and animals. So they are considered sometimes as fungal elements. Fungal animals. They are resembling protozoa because they are performing the amoeboid type of movement. Amoeboid type of movement means with the help of pseudopodia. Pseudo means falls. Podia means legs or appendages. So basically that are not the true legs, but false legs helps in the movement of the animal, and it resembles fungus because they are going to produce spores. They are going to produce spores. That is why they are called fungal animals. Fungus-like character and animal-like character. Now, they do not possess other characteristic of slime molds. They do not have cell wall. They do not have cell wall, and they cannot perform photosynthesis. They cannot perform photosynthesis because they are achlorophylous. What do you mean by achlorophylous? That it do not have chlorophyll pigment. So they cannot perform the function of photosynthesis. So as far as their nutrition is concerned, they are saprophytic or saprozoic. That is, they are saprobes. What are saprobes? That these saprobes are absorbing their nutrition, from dead and decaying organisms. As we have earlier discussed, what are saprobes? They can be saprophyte or they can be saprozoic. So saprophytic or saprozoic. Saprophytic means when they absorb nutrition from dead and decaying plants. Saprozoic when they absorb from dead and decaying animals. So these saprobes absorb nutrition from dead and decaying organism. So these slime molds do not have cell wall and they cannot perform photosynthesis because they do not have chlorophyll pigment. So these slime molds can be of two types. These slime molds can be of two types. Number one, acellular. Acellular slime molds and cellular slime molds. Acellular slime molds and cellular slime molds. So first we are going to discuss about the acellular slime molds. Acellular slime molds. Now, acellular slime molds are basically in the form of slimy patches, and these are found where they are found? They are found at shady, moist places, and they are generally found on dead and decaying organism. So they are saprobes. They are absorbing nutrition from dead and decaying organism. And these acellular slime molds under unfavorable conditions shrinks and forms a covering, a cyst-like covering, that is for the perennation. and that structure is known as sclerotium. Under unfavorable condition, the plasmodium shrinks and forms sclerotium for the purpose of perennation. Now, as far as its perennation is concerned and increase in the number is concerned, it produces spores with the help of the organ in which spores are present are called sporangium. So this plasmodium, which forms the sclerotium during unfavorable condition, when under favorable condition, the plasmodial mass undergo cleavage, that is, it divides and forms spores like structures. These spores forms cells called swarm cells or non-motile mixed amoeba. So these swarm cells fuses and forms zygote, and this zygote will ultimately forms a new plasmodium. So let's have a look with the help of a diagram. So say this is a plasmodium because it do not have any fixed structure. It is many nucleated. It is basically the association or colony. This will form a sporangium like structure, say like this. This is sporangium. This sporangium will gives out spores. These are spores. And these spores will burst and form flagellated swarm cells. These are flagellated swarm cells, and sessile or non-flagellated cells called mixed amoeba. These will join with each other at the non-flagellated end. This will fuse with each other and they can form, from both the roots, they can form the zygote. And this will lead into the formation of a plasmodium. So this is the cycle. This is the haploid phase and this is the diploid phase. These are haploid phase and this is diploid phase. This is diploid. haploid and diploid. That is alternation of the generation. This plasmodium will give rise to sporangia. Sporangia contains spores. These are released during favorable condition. This will form two types of cells, swarm cells and mixed amoebae. Swarm cells will fuse with each other and mixed amoebae also will fuse with each other and forms the zygote, and these zygote will form a new plasmodium. This is the overall cycle of the acellular slime molds. Then we have next type, that is cellular slime molds. Cellular slime molds. These cellular slime molds are basically uninucleate haploid cells. Uninucleate haploid cells which divides to form many haploid cells called mixed amoebae. That is called mixed amoebae. Mixed amoeba or mixed amoebae. These amoebae divides to form many haploid cells called mixed amoebae, and this mixed amoebae forms cyst that is covering over the mixed amoebae during unfavorable condition. Unfavorable condition, that is known as microcyst, which will germinate when favorable condition prevails. Now, when this is also known as community slime mold. Why it is called community molds or community slime molds? There is this interesting phenomena, community slime molds. Community slime molds. Why these are known as community slime molds? Because during the condition of exhaustion of food, that is when there is no food in the vicinity, then they will join with each other. The uninucleate unicellular slime molds will unite with each other and forms a plasmodium like, not plasmodium, the plasmodium like structure. That is why it is called pseudoplasmodium. Pseudoplasmodium. So plasmodium in acellular type, in cellular type it is forming pseudoplasmodium, and this pseudoplasmodium will move with the help of pseudopodia. Pseudopodia, that is false legs. Pseudopodia, and absorbs nutrition with the dead and decaying organism. That is, they are saprobes. They are also saprobes. So, after getting the nutrition, it gives out sporangia, which gives out spores, and that germinates into new mold that absorbs saprobes nutrition by saprophytic nutrition, and sporangia under favorable condition it forms sporangia, and sporangia contains spores. It will be liberated and forms new mold, and it, apart from this, it also forms a structure known as sporocarp at the front end. The sporangia are found at the rear end. So as they form the microcyst, sometimes what happens in the pseudoplasmodium? What is pseudoplasmodium? It is the aggregation of the mixed amoebae. It is the aggregation of mixed amoebae. So sometimes what happens, the mixed amoebae of pseudoplasmodium, mixed amoebae of pseudoplasmodium, what they do? They engulf their adjacent, mixed amoebae and forms zygote by karyogamy. What is karyogamy? Gamy means fusion, karyo means nucleus. There will be fusion of nucleus and forms the diploid zygote that will result in the formation of new cells after meiosis. So this can be seen with the help of a diagram, an indicative diagram. Say this is the pseudoplasmodium. This gives rise to sporangia. This sporangia gives rise to spores. These are spores. There will be germination of spores. This causes germination. This is sporangia. This is pseudoplasmodium. And this will be the generation or germination of spore. This is spore, and that can give rise to pseudoplasmodium with the aggregation of mixed amoebae, and they can result in the formation of mixed amoeba. So this was all about the slime molds, acellular type and cellular type. Then we have the protozoa and protists like our amoeba gingivalis, anti-amoeba gingivalis, that can be dealt separately in the protozoa file. So what we have seen in the kingdom protista, that all those organisms with single cell but eukaryotic organization, that is having the membrane bound organelles and 80S type of ribosomes. Then we have seen, what we have seen, the locomotor organs, they can be locomoted by the flagella, cilia or pseudopodia. So we have divided different types of protistans into different groups, that is dinoflagellates, diatoms, then we have euglenoids, then we have molds, slime molds, and they are interesting because they are considered as the connecting link between the animals and the fungus. Euglenoids, that is euglena-like organism, they are also interesting because they are the connecting link between the animals and plants. So this protistan kingdom comprises of very interesting creatures which can be a boon to the evolutionary, or which can focus on the evolutionary trends of the organisms, that which organism is evolved from which type of organism. So kingdom protista includes dinoflagellates, diatoms, euglenoids, as well as slime molds and protozoan protists. So this is all about the kingdom protista. Thank you.